go. And I even have a post-it note, and it's still not helping. Okay, so again, let me repeat that. Okay, so you have two labs, okay, for today's lab. Okay, one is a karyotype lab. That one you have to complete, okay, for a grade. Any points you lose count against you. Any points you gain definitely help you. All right, that one has to be submitted if you want, if you don't want to get a, how do you say, lose a grade. However, the second lab, Patterns of Inheritance lab, that is a bonus lab. You do not have to do it. Okay, it's up to you though. It's worth 20 points. Okay, that doesn't mean you'll get 20 for doing it. It just means you have the opportunity to gain extra points. So please keep that in mind. There are two labs. Okay, one is mandatory, the karyotype, and then the other one is just a bonus lab. It's up to you whether you do it or not. They're both due a week from today at the usual start time for lab, okay, 9:30, preferably before. Now, the first lab, the karyotype lab. Okay, first thing I want to say about the karyotype lab. You do not have to put the answers into the Word document or convert the Word document to PDF, put the answers in. Listen carefully. If you want, okay, what you can do is for the answers for the karyotype lab, you can type them into a separate doc. Just give me the page number if it's a karyotype, and I'll tell you what's required for the karyotype that you're identifying. And then there are summary questions or review questions at the end. You can type those on separate doc also. Just give it a title so I know it's your final set of questions on the last page of the lab, and you can just type the answers. So again, for the karyotype lab, you don't have to put your answers into the Word document. You can type your answers in a separate doc. Just make sure I know what page the answers are coming from, and then you can submit that instead. Any questions about that? Okay, so let's go forward with the first lab, karyotype lab. Okay, we talked about a karyotype. It's a chromosome map. Okay, so a karyotype is a way of identifying characteristics of the chromosomes of an individual by taking a photomicrograph of chromosomes from a single cell and then arranging the individual chromosomes into pairs. So we've talked about this before. Technically, they're pairs of sister chromatids because they remove the chromosomes from cells that are in the process of undergoing cell division involving mitosis. But I posted a video. Oh boy, here we go. Okay, I noticed a little hand didn't move, so hang in there with me. So, but I believe I posted a video, I hope I did, for the amoeba sisters where they explain since sister chromatids, since each chromatid is connected at a centromere, for the sake of simplicity, okay, when we talk about a karyotype, since they're still attached to each other, these sister chromatids, we just say chromosomes for a karyotype, okay? So we simplify it. We talked about a karyotype demonstrates how there are 23 pairs of chromosomes, okay, in our somatic diploid cells of the human body. The first 22 pairs are called autosomes. 23rd pair, they're called the sex chromosomes. They have to do with determining the sex of the offspring. Okay, when they perform a karyotype, if the karyotype looks something like this, it's telling you two things. First, that the offspring appears to be normal, 23 pairs. Second, it's telling you the sex of the offspring. So listen carefully. You're going to have karyotypes to identify, okay? If the karyotype is of a normal person, you'll just simply put down as part of the answer normal. But then you'll give me a second part of the answer. The second part of the answer will be, is it the karyotype of a male or female? Now, you will be given karyotypes that are abnormal. In that case, you will give me the condition they have, such as Turner syndrome, and you will tell me what sex, okay, is the karyotype of, male or female, okay? So that's what you'll do for the answers on the karyotypes. Let me jump ahead. So when they do karyotypes, they take cells that they catch dividing through a process that includes mitosis. So they're basically catching the cells during metaphase of mitosis, and they extract the chromosomes, and that's when they're easiest to pair together, creating the karyotype. It gives you some information on the first page. Now listen, you do have to read through this lab, okay, because a lot of the information they give you is to help you answer the questions at the end of the lab. Some of those questions at the end of the lab also are thinking questions, okay? So one thing I'm seeing with lab reports is there are those students who understand I have to study the information so I know what answer to give, and then I have that other group of students, okay, turning in their lab reports on time. They're answering all the questions, but it's pretty obvious. They're just trying to throw an answer down and get it over with as quick as possible. those grades aren't going to turn out well. You actually have to put some effort into this, everybody. That's the trade-off. Lab is normally 2 hours and 40 minutes. It should take you quite a bit of time, okay? Now, however, some of the karyotypes that are going to be given to you, you should be able to figure out the sex, such as if you get a karyotype that has XY, you know it's male. You look at the 23rd pair to determine the sex. Or if it's XX, that tells you it's female. Now, the way it's going to work, and it's not shown here, if you're given a karyotype that has a Y chromosome present, and it's shown to you there's a Y chromosome present, assume the karyotype is from a male. If you're given a karyotype that only has an X or X chromosomes, but there's no Y present, assume the karyotype is female, okay? Now, the next thing you'll look at is normal number of chromosomes. If you look at this karyotype, first of all, it tells you it's male. And if we look, as far as we can tell, there are 23 pairs of chromosomes. However, if you look at this karyotype, initially it looks like the autosomes are all paired together. I pass by the sex chromosomes, they're paired. But when I get to the 21st pair of autosomes, notice there's not a pair. They're showing you there's 3. In that case, you would write down what does this mean in terms of what type of syndrome or disease does the individual have. Okay. This one here is known as Trisomy 21, but it goes by something more popular. You can give me any name. And then over here, you would tell me whether it's male or female. Now, if it's an abnormal karyotype, meaning it has either too many or too few chromosomes, that's due to a process called aneuploidy. Okay, and you're responsible for knowing what aneuploidy is. Aneuploidy, first of all, what causes this? It's caused by nondisjunction during meiosis. Okay, in other words, how is it we could end up with a karyotype where we don't have 23 pairs of chromosomes? In other words, this person does not have somatic diploid cells. These cells are not diploids because they have one too many chromosomes. Often it's written down as 2n+1 to denote, hey, there's an abnormality here. Well, what causes this abnormality? It's called a process called nondisjunction, and I believe I have a video link for it. What happens is either during meiosis 1 or during meiosis 2, okay, segregation or separation doesn't occur. During meiosis 1, sometimes what will happen is that when the tetrads form the metaphase plate, one of the tetrads, the homologous pair that forms it, they're not segregated. Instead of one going this way like the top ones here and one going the other way, what happens is a homologous pair that forms a tetrad is not segregated and they both go in one direction. If this happens during meiosis 1, nondisjunction, then 100% of the gametes produced will be aneuploidy, meaning then that the offspring will be aneuploidy. What do we mean by aneuploidy? We don't have the correct number of chromosomes. In the case of the gametes, there are either one too many or one too few chromosomes, denoted as n+1 or n-1, or if it's in the offspring, we might put down 2n+1 or 2n-1 to denote, okay, that we have a condition called aneuploidy, an abnormality. However, sometimes nondisjunction occurs during meiosis 2. So what happens is we get segregation of homologous pairs going from a diploid nucleus to 2 haploids. But the purpose of meiosis 2 is to separate the sister chromatids into daughter chromosomes. And sometimes what happens is in one of the 2 haploid nuclei that are going through meiosis 2, in one of the 2, there's a sister chromatid that's not separated. In the other nucleus, everything goes fine. So in this case, if nondisjunction happens during meiosis 2, 50% of the gametes produced are not haploid. They're aneuploidy. They're either carrying one too many or one too few chromosomes. 50% are normal. They're true haploid gametes. Okay. Well, if any of the gametes that are aneuploidy, that are carrying an incorrect number of chromosomes, are involved in fertilization, that can result in an abnormality that is revealed when a karyotype is performed, such as this Down syndrome known as trisomy 21. It's the most common cause of Down syndrome, but it is not the only cause. Make sure you know that, because that's also part of what you're going to need to know. Okay, so this individual here, now it's showing it's trisomy 21. It has nothing to do with the sex chromosomes. So the offspring could be male or female because it affects one of the autosomes. Okay, but this is the most common cause of what you know as Down syndrome, but geneticists call trisomy 21. It's due to nondisjunction causing aneuploidy. There are also forms of aneuploidy that affect the sex chromosomes. It's known that there are males out there, they tend to grow taller than average, but they're sterile, and they're found to have, an abnormal karyotype, due to it meaning they have an abnormal number of chromosomes. We call this Klinefelter syndrome. Listen carefully, okay? With Klinefelter syndrome, first of all, the offspring is always male. So notice, it's showing you here, okay? There's always a Y. So when it comes to Klinefelter syndrome, it's a type of aneuploidy, but it only occurs in males. Second, it's not showing you everything over here. It's just showing you where the aneuploidy has occurred, okay? Where the incorrect number of chromosomes are located. For Klinefelter syndrome, this is where a male inherits too many X chromosomes. So if you look at a karyotype, the first 22 pairs of autosomes should be normal, all pairs. But when you get to the sex chromosomes, instead of there just being an X and a Y, you could end up having 2 or 3, X chromosomes and a single Y. That lets you know it's Klinefelter syndrome. The autosomal pairs are all fine, the first 22 pairs, but when I look at the sex chromosomes, I see a Y, so I know it's a male, but there are too many X chromosomes. Here they're only showing you 1. It could be 2 extra or 3 extra, okay? That's Klinefelter. There's another condition. They don't have it here, but you can write it down. It says XYY. Okay, this is another form of aneuploidy affecting the sex chromosomes. They actually call it XYY syndrome. In this case, again, I said if you see a Y present, assume it's male, and in this case, it's a couple of extra Ys that the male inherits, but notice over here it says they're normal. Sometimes in females, you'll find 2 different types of aneuploidy, okay, that have to do with either inheriting too many X chromosomes or being short one. So over here it shows you there's a type of aneuploidy that affects the number of sex chromosomes. So it's a sex chromosomal disorder. They show you the female inherits 3 X's. It doesn't say it here, but I want you to know it as triplo X. And this is in your notes also, triplo X syndrome, okay? So I said there's no Y chromosomes here, so we assume they're female, but it just so happens women, that have this type of aneuploidy called triplo X, they're normal. And there's more to it as to why. I'm not going to go into it, but there are reasons why. But then if you look at this one, there's another type of aneuploidy that you only find in females, and it's called Turner syndrome. This is where a woman, every cell of her body is not diploid. They're 2n-1. Her somatic cells are 2n-1. When you do a karyotype, the first 22 pairs of autosomes are all there, but when you look at the 23rd pair, the sex chromosomes, the female has the offspring. The female offspring has only inherited 1 X chromosome. The 0 means they didn't inherit a second sex chromosome like they should. In other words, they don't have a 23rd pair. These offspring, notice there's no Y, so they're always female, but what they're not showing you is that women with Turner syndrome are often sterile. Okay, so when you look at the karyotypes, make sure you indicate whether they're normal or abnormal. If they're abnormal, figure out what type of aneuploidy they have. And you do that by first locating, is it in the autosomes that there's an incorrect number, or is it in the sex chromosomes? If it's the sex chromosomes, here's a table of choices, but I told you to give these 2 specific names, XYY syndrome, triplo X. And then also give me the sex of the offspring. Okay. So answer all questions in the, did I say lab 8? Should be lab 9, everybody. I'll fix that. Lab 9, that's a typo there. God almighty. It's a typo. Okay, and it'll be due a week from today. Okay, remember, the Karyotype Lab, that's a mandatory lab. You need to turn it in for a grade. You don't want it to hurt your grade. You want it to help your grade. And remember, for that Karyotype Lab, you can type the answers up on a separate document. Just give me the page number so I know where the answer goes to. Keep it organized, okay? Or you can just insert it into the document I gave you, but it'd probably be easier if you just put the answers separate on a Word doc, okay? Answer the questions also on the last page, please. And don't forget for the karyotypes, You need to let me know if they're normal or if they're abnormal. And if they're abnormal, let me know what type of syndrome it is. And then also let me know the sex of the person, okay, that they used to create the karyotype. And then the final questions at the end, that's where you're going to need a lot of the background information that's built into the doc. But the answers are there except for a couple of thinking questions. Okay? All right. And I'll fix that PowerPoint. It should be lab 9. I'm sorry, everybody, in my haste. Okay, the second lab, that's a bonus lab. The second lab 9, that's a bonus lab. It's a patterns of inheritance lab. Okay. This pattern of inheritance lab, if you decide to do this bonus lab and submit it, you can also type it up, okay, and put the answers on a separate document. Now, there are Punnett squares that you have to fill in. If you say you use a Word document, you can just go to tables and create the Punnett square using tables and then fill it in with the necessary information. Okay, so patterns of inheritance is a second lab, and it's a bonus lab if you want to do it. Now listen carefully. On page 1, I forgot to delete it, but on page 1 of this bonus lab, patterns of inheritance, you don't have to answer the questions about the coin tossing if you don't want. Okay, it's not going to affect you one way or the other. Okay, so basically you're going to start answering questions around page 2 or 3. But it's patterns of inheritance, and you're going to be asked to fill out Punnett squares in relation to recessive genetic disorders, dominant genetic disorders, X-linked genetic disorders, as well as filling out Punnett squares having to do with your ABO blood type. I believe I have a couple of links in that document so you can watch the videos to help you out. Now, when you do the Punnett squares, okay, you can do them one of two ways. Normally, the way I do it is not the way it's shown here. Normally, what I do is when I do it in a Word doc, is I'll go to tables and I'll ask the table to give me 3 rows and 3 columns so that this information having to do with the sperm and the egg, it's also contained within boxes. Okay, but then I make it clear in the boxes, I just put down the allele. You don't have to put down the circles, you can just put down the allele. So the gametic information from the parents goes in the top edge or the side edge. And doesn't matter. Okay, the female information can go here or the female information can go here. Sperm can go here or here. Most often it's arranged this way, but I've seen it arranged the other way. The bottom line is make sure the gametic information, the alleles carried in the gametes, specifically the allele information or genotype is put here. Then from there with the Punnett square, like I said, it's about probability. There's a probability that this sperm carrying this allele will fertilize this egg carrying this allele. I end up with a genotype that's homozygous dominant and a phenotype that's purple, and you just keep carrying it out. Then it's going to ask you questions. It'll ask you questions about the phenotypes, and it may ask you questions about the genotypes. Remember, phenotypes, physical appearance, observable feature, such as for this cross, we have a 3-to-1 phenotypic ratio, 3 dominant purple to 1 white. If it asks you about genotype, well, here they're showing you that in this case, the Punnett square has a genotypic ratio that doesn't match up with the phenotypic. It's a 1 to 2 to 1. 1 homozygous dominant, 2 heterozygous dominant, 1 homozygous recessive. So you'll be asked to do different Punnett squares associated with different conditions. I'm going to jump ahead. Okay. So the Punnett squares you'll be doing, many of them will have to be associated with dominant versus recessive genetic disorders in the beginning. So I believe one of the first Punnett squares you're going to fill out has to do with cystic fibrosis. This is a recessive genetic disorder tied into a recessive gene mutation or a recessive allele, and it gives you some background info here. But you only have to answer the questions that are in the lab. Another disorder that you'll be filling out a Punnett square for is Huntington's disease. Okay, so let me backtrack here. For cystic fibrosis, I simply use an uppercase C or a lowercase c to represent the 2 alleles for cystic fibrosis. Okay, the dominant allele, the uppercase C, that's the normal allele. Therefore, if the offspring inherits just 1 uppercase C allele, they should be normal, but they might be a carrier. Now, if they're a carrier or they have the recessive genetic disorder called cystic fibrosis, then that means there will be a lowercase c allele because it's a recessive allele that causes this recessive genetic disorder. If they have cystic fibrosis, they have to be homozygous recessive. But they could be a carrier, meaning they're heterozygous dominant, but they're carrying that hidden trait in the recessive allele for cystic fibrosis. It will ask you questions going back and forth on this, but it'll ask you to fill out a Punnett square, and then it'll ask you questions such as, if I can find it here, it'll ask you questions such as, okay, if both parents are carriers, using a Punnett square, what's the probability that the child could be born that is, say, has cystic fibrosis? Here it says deaf, but it's recessive, and you would say 1 in 4 or 25%. What's the possibility that they would be normal and not a carrier? 25%. What's the possible that they might be a carrier? Well, 2 in 4 or 50%. So you can give me answers in fractions or percent when it asks you those questions. Okay, but make sure you differentiate between when it's asking phenotype versus genotype. Now, Huntington's, what I use for Huntington's is an uppercase H or a lowercase h for the allele. Okay, listen carefully. The uppercase H allele, okay, the dominant allele, it causes a dominant genetic disorder known as Huntington's disease. The lowercase h allele, that's the normal allele. So in order for a person not to have Huntington's disease, inherit this dominant genetic disorder called Huntington's disease, you have to be homozygous recessive to be normal. What geneticists found out is that for dominant genetic disorders, the offspring only has to inherit one dominant allele, and they will have that genetic disorder. You will see it in their phenotype. It's just a matter of when. So down here, remember I told you at the end of lecture, Huntington's is a ticking time bomb. It's scary. Because unless you have a pedigree together or just happen to just notice in the family that several members have this genetic disorder, okay, you'll reach your 30s to your 40s before you start seeing changes in your phenotype. And like I said, once those symptoms show, you roughly have 5 years. I've seen some documentaries on this, and you talk about a nightmare. Okay, so but the catch is this: when it comes to dominant genetic disorders, if the offspring inherits a dominant allele from both parents associated with Huntington's disease, the mother will miscarriage. So when it comes to dominant genetic disorders, the only offspring that survive to birth are heterozygous dominant. So please remember that, okay? For Huntington's disease, those that are diagnosed with this dominant genetic disorder that live to about 30 or 40 before symptoms show up, they are heterozygous dominant. But it's the dominant allele that causes this dominant genetic disorder. Then we have sickle cell disease. I cheated. It falls under what we call incomplete genetic disorders, but I want you to think of it as a recessive disorder. But the reason why they call it incomplete is because first of all, it's more of a recessive genetic disorder, okay? It's a recessive allele that's associated with causing a genetic disorder called sickle cell disease, okay? If you inherit 2 of these recessive alleles, 2 of these mutated genes, you will have sickle cell disease. However, the reason why sometimes they don't say this is a recessive disorder, the reason why sometimes they'll say it's incomplete, is because for individuals that are carriers, individuals that are heterozygous dominant, okay, the dominant allele, which I use an uppercase S, that's the normal allele. For those that are carriers, they'll inherit a dominant allele, uppercase S allele, and a recessive allele, a lowercase s allele. They're carriers, these individuals, but under certain conditions in the environment, okay, that recessive allele could have an impact on their phenotype. So these individuals are sometimes referred to as having sickle cell trait, but a person with sickle cell trait is a carrier. They're often normal, and something in the environment, like a change in air pressure, has to occur for that recessive allele to really have an impact on their phenotype, okay? Versus a person who has sickle cell disease, they're homozygous recessive. They've inherited 2 recessive alleles. Both those recessive alleles cause a recessive genetic disorder called sickle cell disease, and from birth, the offspring is having problems. Lifespan used to be roughly up to 25 years of age. So many of the genetic disorders, they appear at birth. Now, other genetic disorders are what are called sex-linked genetic disorders, but more specifically, X-linked, such as the genetic disorders hemophilia and colorblindness. These two genetic disorders are tied into alleles found on the X chromosomes. Therefore, instead of calling them sex-linked, often geneticists will more specifically call them X-linked disorders. Okay? There's an allele. It's actually a recessive allele in females, okay, associated with causing hemophilia. Women inherit a pair of X chromosomes. So let me go over here. A pair of X chromosomes, okay? The only way they could have hemophilia, all right, is if both X chromosomes, so if you look over here to the right, the only way they can inherit this genetic disorder known as hemophilia, or in lay terms, a bleeder's disease, is if they are homozygous recessive. And the way we denote, okay, any alleles that are located on sex chromosomes and are associated with causing genetic disorders, the way we represent them is we'll put the letter for the sex chromosome, and then the allele will be put as a superscript. That's different than the way you'll name the allele for, say, sickle cell or cystic fibrosis or Huntington, where you just use an upper and lowercase letter. In this case, if the genetic disorder is tied into an X-linked gene or X-linked trait, you'll give the X chromosome, and then the allele will be put as a superscript. And it'll either be an uppercase or a lowercase. Okay, for hemophilia in women and for color blindness in women, it's a recessive genetic disorder, or they're recessive genetic disorders. The only way a woman will have hemophilia or the genetic disorder for color blindness, they have to be homozygous recessive. On both X chromosomes, they have to inherit the allele for color blindness or hemophilia. Now you can use, again, uppercase C, lowercase c, uppercase, what do we call H, lowercase h, because they're going to be superscripts next to the X chromosome. So there shouldn't be any confusion when compared to sickle cell or Huntington's. Is that clear to everybody? Okay. Now, in men, for men, we are what we call hemizygous. Hemi meaning half our sex chromosomes are X, half our sex chromosomes are Y. So for males, okay, when it comes to hemophilia and color blindness, you primarily find these genetic disorders in males. Because for males, if you look over here to the left and over here to the right, notice they're showing you the X chromosome, but the superscript contains a lowercase letter. In males, what is a recessive allele functions as acts as a recessive allele in females. In males, it doesn't work that way. Okay? There's a lot of genetic information on the X chromosomes that has nothing to do with sex. Hemophilia and color blindness are perfect examples. There is a clotting factor gene located on the X chromosome. Okay? The dominant allele is the normal allele or normal gene for clotting. And this gene codes for a clotting factor that helps you form a blood clot. However, there is a mutated form of this clotting factor gene located on the X chromosome, which is recessive. Well, what happens is, for women, if they're heterozygous dominant for this clotting factor gene, in other words, they inherit one normal clotting factor gene, but one mutated recessive one, that one normal they inherit is enough for their body to form blood clots whenever they get it cut. But however, for males, down here, we only get one copy of the genes located on the X chromosome. So if we inherit, say, the mutated, the recessive allele for the clotting factor gene, there's no backup X chromosome with a backup X allele in normal males. Therefore, you will see the genetic disorder hemophilia in males. And it follows the same pattern for color blindness. There's a gene that codes for proteins that allow you to differentiate between different colors. That's located on the X chromosome. The dominant allele is a normal gene. So for a woman, if she's just heterozygous dominant, she'll have normal color vision. But for males, we only inherit one X chromosome. So if we inherit the mutated version of that gene, the recessive allele, then we don't have the ability to differentiate between at least certain colors and we are therefore colorblind. Okay, so you have, when you go over that, notice the difference between the two. Now for blood typing. Blood typing here, I posted a video. Watch the video. For blood typing, ABO blood typing is what you're going to be doing, filling in Punnett squares for and answering questions. But you have to know this. There are not a pair of alleles for blood typing. There are a total of 3 different alleles, and 2 of those alleles are dominant alleles. Therefore, if you come down here where it talks about phenotypes, if a person's blood type or phenotype is type AB, what that means is they've inherited a dominant allele from one parent and a dominant allele for the other parent, but these dominant alleles code for different carbohydrates, or what we call antigens. However, both dominant alleles equally impact your phenotype. They both contribute equally to your phenotype. Therefore, these 2 dominant alleles, we talk about how they exhibit codominance in your phenotype. So a person that has blood type AB, the allelic pairs, their genotype is dominant A and dominant B. Now the third allele is a recessive allele. We use a lowercase I. So if you hear somebody has a phenotype, blood type A, that means they're homozygous recessive. Now if the offspring has blood type A or separately blood type B, come up here and notice there's 2 different genotypes the person could have. Person with blood type A could be homozygous for dominant A or heterozygous for dominant A. A person with blood type B could be homozygous for dominant B or heterozygous for dominant B. So the medical community and the research community realized there was a time period where, okay, we took blood from mom or dad and gave it to the son, and when we did it the second or third time, there was an adverse reaction. Well, it is possible for the offspring to have a different blood type than one or both parents. Okay, so when you do the Punnett square, watch the video. Keep in mind there are 3 different alleles that are responsible for, notice, a total of 4 different phenotypes. Okay, and I'll let you look at that there. Again, okay, I fixed this one. I didn't fix the other one. So for lab 9, this is a bonus lab. You don't have to do it. It's up to you. You can do it on separate paper and only give me the answers, but put the page numbers. But wherever you have to do a Punnett square, redraw the Punnett square. And like I said, you can add in extra boxes so you can put in the information having to do with what alleles are being transferred by sperm or egg. Okay? But you can redo the Punnett square, and like I said, you can do the answers on a separate sheet of paper if you want, or separate word, I'm sorry, type them in and a different doc, and then just use the table format if you're saying Word doc. I have a question. Did you want the fill out Punnett squares? Yeah, that's what I'm saying. Redraw the Punnett squares. If you're doing Word, just go up to tables, and then fill it in with the genetic information. Does that answer your question, Desiree? Okay. Remember, it's a bonus lab, okay? So, but if you do submit the bonus lab, make sure you try to complete it, please. All right. Are there any questions? Okay, I'm going to